Hello and namaste everyone. Welcome to Journey to Wilderness podcast, the voice episode मा र आज पनि हाम्रो voice episode मा German herpetologist and researcher Kristin Mahlow लाई लिएर उपस्थित भएका छौं र विगतमा जस्तै हामीले हाम्रा गेस्टहरुको चाहिँ प्रोफाइलहरुको बारेमा यहाँलाई जानकारी गराएर उहाँहरुसँग हामीले सोधिएका प्रश्नका चाहिँ उत्तर सहितको भिडियो चाहिँ यहाँहरुमाझ प्रस्तुत गर्दै आएका छौं र आज पनि हामीले herpetologist and researcher Kristin Mahlow सँग केही प्रश्नहरु सोधेका थियौं र त्यही प्रश्नको चाहिँ उत्तर लिएर यहाँहरुमाझ उपस्थित भएका छौं र विशेष गरी म अलिकति के जानकारी गराउन चाहन्छु भने विदेशी रिसर्चर साइन्टिस्ट herpetologist कन्जर्भेसनिस्टहरु उहाँलाई बोलाएर हाम्रो यस जर्नी टु वाइल्डनेस पोडकास्ट द भोइसमा लिएर आउनुको उद्देश्य चाहिँ उहाँहरुबाट चाहिँ हामीले एउटा एजुकेशनल पर्पजको हिसाबले केही कुराहरु सिक्न सक्छौं हाम्रा नेपालमा पनि साइन्टिस्टहरु रिसर्चर्सहरु धेरै हुनुहुन्छ त्यस्तै विषयका स्टुडेन्ट्सहरु पनि धेरै हुनुहुन्छ उहाँहरुलाई केही मात्रामा एउटा आइडियाज एउटा नलेजको चाहिँ एउटा शेयरिङको एउटा प्लेटफर्म दिन खोजेको अथवा बनाउन खोजेको भनेर यहाँहरुले बुझ्दा हुन्छ सो बिफोर वी स्टार्ट अबाउट द भिडियो अफ हर म चाहिँ उहाँको बारेमा थोरै भन्न चाहन्छु Kristin Mahlow is a German herpetologist and city imaging specialist at the Museum of Naturkunde in Berlin, part of the Leibniz Institute for Evolution and Biodiversity Research. She currently serves as a scientific research assistant while pursuing her PhD with a focus on cranial morphology of snakes, particularly venomous and colubroid species. Combining herpetology and advanced imaging techniques, her work contributes to deeper understanding of snake evolution, anatomy, and biodiversity. उहाँले आफ्नो PhD गर्ने क्रममा उहाँले चाहिँ केही नयाँ कुराहरु चाहिँ एउटा यो स्नेक मोर्फोलोजी ग्ल्यान्ड मोर्फोलोजी भन्छ त्यसको विषयमा उहाँले के कुरा पत्ता लगाउनु भएको भन्ने कुरा पनि हामीले चाहिँ म्युजियम अफ नाटुकुण्डेको वेबसाइटमा रिसर्च गेटमा पाउँछौं र के पत्ता लगाउनु भयो त्यतिबेला भन्ने कुरा चाहिँ म थोरै जानकारी गराउन चाहन्छु key findings and collaboration highlights. Mahlow's PhD project in collaboration with University of Sao Paulo examines how venom gland structure varies from juveniles to adults in Bothrops jararaca. She found that gland cell arrangement, size, and spatial configuration change markedly during ontogeny, insights that may impact the development of antivenoms. And broader contribution via CT scanning. Her lab technical leadership has enabled new methods for imaging both hard and soft tissues, pivotal for studies across vertebrate evolution and anatomy, including muscle structure, changes with limb loss in amphibians. Partnership in large-scale morphometric studies. Mahlow contributed to high-density cranial morphometric projects. For example, she supported data acquisition in PNS study, analyzing skull shape diversification across 200 square meter species, demonstrating preserved trait integration patterns underlying broad ecological variation in lizards and snakes. She is also acknowledged in BMC biology work exploring full skull roof microanatomic convergence across reptile lifestyle, underscoring her role in city-based data processing. Publication and sample works, annotated checklist and morphological key to dwarf snake, genus Ironis, with emphasis on definition, vertebrate zoology, co-authored work on colubroid snake morphology and virtual segmentation, and contribution to integrative publications such as studies on limb, reptile myology, amphisbaenia, revealing musculoskeletal decoupling via CT imaging, systematic taxonomic revision, and snake identification. Example, Storn Hybidium temporale with detailed anatomical support. So अहिलेलाई चाहिँ मैले उहाँको अझ यो धेरै नै छ उहाँको प्रोफाइल, तर मैले छोटोमा त्यति भने. So we have asked her some questions about her work and about her research. So without delaying, let's hear her answers. Thank you. Keep watching. What are the capabilities and application of the CT facility at the museum for Naturkunde, and how has it advanced research in comparative anatomy?
So in 2010, we started developing a CT lab in the National History Museum in Berlin. And the good thing with CT technique, you maybe know it from broken a leg or something, is that you go to the hospital normally and then they make an X-ray and they see how it is broken. And in 2010, we decided that this is also a good technique for scientific reasons. So for my master thesis, I went to the Humboldt University and I get contributed to this team working with CT techniques. And for my thesis, I wanted to analyze dwarf snakes, so small snakes, in comparison to the big relations. So the question was, if when I have a small snake, what happens to the skull itself? Is it just stop growing in comparison to the bigger species, or do they do something completely cruel? And for this analysis, I wanted to analyze around 700 to 800 specimens, and I want to make measurements and the skull itself. In former times, it was done by wrapping off the skin, wrapping off the muscles to see the bony structures. But of course, for 700 specimens, nobody would have allowed me this study, right? So I cannot destroy 700 specimens to figure out, okay, nothing happens. So that's the reason why we started working with CT scannings. And the technique is quite easy. So you make an X-ray picture from a certain position of a skull, and then the skull turns around in 360 degrees, and I make always X-ray pictures. In total, I make around 1,400 to 2,000 pictures. And then there's a special software that put together a 3D model. So in our computers, we now have a 3D model of the skull without destroying the specimen itself. And that means I could analyze my 700 specimens, take my measurements virtually, and then I can make my statistics and analysis and stuffs. And this was the start of my career, as it was a really good study of the whole genus Irinus. So it's coming from Turkey to Iran, Iraq, Pakistan. And I wanted to know what are the different lineages doing. So it's around 17 species. And we figure out that there's completely different shapes of the skull, and they completely are adapted to their lifestyle. So it was a good study, and because I was so trained with the CT itself, my supervisor at this time said, hey, do you want to stay and do this also for other research projects? So we opened the lab not just for snakes and lizards, what we did formerly. We started CT scanning insects, fossil specimens, and then at one point we realized that our machine is too small. So we just could analyze specimens up to 5 centimeter skull size. But I get more interest in bigger snakes, venomous snakes, and how the venom gland is developed and everything. So we buy for the museum a second CT machine. And this means that I can now scan up to 90 centimeter in size specimens. So we have a small one and a really big one right now. And then it comes up to fishes, so I get a lot of projects for fishes. And the fishes are more complicated than all other specimens for preparation for CT scanning, so we buy the third machine. So in total now we have 3 machines that we can use for X-ray and CT analysis. And then more and more projects come up, and a lot of more people thought, hey, cool, nice technique, I not have to destroy something, I can look inside all my characters. Can you scan this and this and this and this? So in the moment I have around 50 projects in parallel, from spiders, small spiders, and little insects, up to mammals, bovid skulls. So these water buffalo skulls are huge. And we want to analyze all of these. And I have around 70 students that I supervise in the techniques itself. So we buy more and more computers, high power computers. So in total I have 3 labs now. I have the CT lab, I have the visualization lab where we have all the computers and students. And then the third one is that we can make also surface scans. So when you go to your National History Museum here, you will see that they also have prepared skeletons. And that means I do not need necessarily a CT scan because I already see the skull. And we digitize these with surface scanners. So just capture the surface itself. And it improves the scientific world in this sense that during the last 15 years we developed the techniques, especially for scientific purposes. Former times it was medical scanners, and now we have certain scanners just for us. And when I have digitized a specimen, I can send it as often as I want to the people outside in the world. So in the moment we have projects in Tanzania, Africa, we have projects here in Nepal, of course, and we have other projects in America, whatever. I not have to take the specimen out of the jaw in a box and send it to Africa so that the science can be done there. I just make a CT scan and send it digitally. So this is good first for the specimen because it not has to be shipped. Second, it's good for the scientists because they not have to travel to Berlin. They can stand in their home place. And last but not least, I can copy this data as often as I want. So I can spread the data throughout the world. In case we have a server crash or something, at least our data are out in the world. And this opens the possibility of projects, right? So I'm working with snakes and I'm interested in dwarfism and venomous snakes, but maybe another colleague say, hey, I'm interested in the vertebrae of the same specimen, or I want to count the teeth. So I can just copy the data and send it back and or for another person so that they can do completely different research or art projects. Or sometimes we want to print these specimens in a huge way for exhibitional stuff or artist stuff. So I do one CT scan and I have millions of possibilities what to do. And this really improves a lot. Yeah, it was also by accident. So as I started my master's, so I'm from the bachelor perspective, I'm a genetic girl. So I do genetics in my bachelor theses. And then I moved to the Humboldt University and I met my supervisor there. And he said, yeah, genetics, see, I'm a paleo guy. So he's normally working with fossils. And I do not have genetic projects so much. Do you want to do morphology? And then I started with the dwarfism of the snakes, and it was my starting point working with snakes itself. And if you see a snake skull itself, it looks so cool, so amazingly how these specimens are built up in the whole skull, how they adapt to their lifestyle. And you easily can tell by the skull what they are feeding, what they are doing in their life, if they climb a tree or if they go in the ground. But the specimens I worked with was colubroid snakes, so really simple bauplan. And venomous snakes, and especially vipers, they have a complete adapted bauplan in their skull. And they have these teeth that they flip out when they open the mouth. Everyone that has seen a spitting cobra or a picture of it sees that the teeth, while open the mouth, comes up. And there's a really high adapted mechanics inside of the skull. And this is really nice to study, and it's impressive how these specimens during their development make these mechanisms possible. And this is why I'm interested in. I'm more focused on the venom gland itself. So the most of the venomous specimens and snakes eat in their lifespan different kind of food. So when they're younger and when they're hatchlings coming out of the egg, they mostly prey on insects. So little crickets, stuff like this. Then they shift when they grew older to cold-blooded prey, so lizards, frogs, stuff like this. And there's a certain point in the lifespan where they shift to mammal food, so mice and all of these little rodents that are out here in Nepal. And I'm interested in what it's doing with the venom. So if I want to eat a cold-blooded prey like a lizard, I have to have a certain venom for it, right? But if I want to eat a mouse, I need a different kind of venom. So what happens to the gland and happens to the venom production systems? Do they change as well? Do they just regrow in a different way? So CT scanning setup itself is quite quite complicated. So you have to know what the scientists want to do. So if I want to scan, for example, a big bovid skull itself, then I have to do a focus on the whole skull. But sometimes I get a request for having just the inner ear or want to see the cavities of the teeth. Then I have to focus on these certain points. So sometimes we do around 5 to 6 CT scans just for one specimen because of the different kind of purposes. And yeah, it's sometimes challenging, but I have to make like a quality check after each CT scan. And if I have troubles, I can also ask colleagues. So there's a big group of X-ray scientists that are spread around the world, and we have like a group where we can chat. And then we say, hey, I have a problem with this in the specimen. So we exchange also our knowledge. So this is quite possible.
So what are some of the most surprising or complex anatomical details you have uncovered using 3D visualization?
As I deal with so many different projects, it's really amazing what scientists can do with our CT scans itself. So there are many stories to tell. One of the most impressive ones that I had is a fossil snake from southern Germany, where we thought by eye that it is a colubrid snake, so one without venom teeth. And then we make a CT scan of it and analyze all the bones. And we realized it's a snake that has venom teeth at the end of the maxilla that you cannot see from the outside, so only with CT scannings. And we put it in a phylogeny and want to analyze it, and realized later on that it's a kind of really specific species that is not existing in Germany any longer, but it's now in Asia and Northern America. So we are currently publishing these to explain the whole story between fossils from Germany, relatives in Asia, but also in Northern America, so that you can see the distribution of these kind of snakes during the world. And this is quite amazing.
So
it's still in public.
You have to do fossils scans also.
Exactly. So we also scan fossil remains, not only for the animals that we have, but also for plants and flowers and human remains as well. There are some that are just described by single bones, so like vertebrae of the whole snake column, which are also in Northern America quite commonly, but we now currently try to figure out what kind of remains we have in Germany and Europe to compare this with these kind of fossil snakes. I'm not familiar with all of the taxonomical stuff like Frank is doing, but I'm more morphologist. So if someone comes up to me and say, do you have analyzed this snake? What is a kind of different to this kind of snakes? This is my work that I'm doing. So I'm not describing new species by my own. I'm just giving hints. So I say, okay, look at the skull bones, they're different, or look at this vertebrae, they are different. This and this could be a character or a change or something for a reason to describe a new species. But I'm not a taxonomist.
But CT imaging could help.
Exactly. Exactly. So for publications that I'm doing together with my colleagues, I mostly make the CT scans and the analysis of the bones. And sometimes it figures out that one species that is commonly known has, let's say, 15 teeth, and the new one that they want to describe as a fresh has 17 to 20 teeth. So these are the hints that I can give. Or I can say, look, this bone is bigger than the other, or it has a different shape. And especially for the teeth-bearing bones, so snake have a lower jaw, and they have a maxilla on the outside, and then they have inside a parietal and a pterygoid bone, so they have 4 tooth-bearing bones per side. And these shapes of these bones and how they are connected to each other give us a hint what they have eaten, for example. So if I have an egg-eating snake, they are looking completely different to ones that eat mammals or to ones that eat fish. And that's what I can say. So I can CT scan them and say, look at these teeth, look at these bones. Because they are structured in these characters, it must be eating mice.
So we can find the differences. We can find, as you say, like differences between egg eating or not egg eating.
Yeah, or
by the structure.
By the structure. So also snail eater snakes have a completely different kind of Bauplan of the whole bones where there are teeth. So you can imagine a snake that eats snails does not need venomous teeth because snails normally not hatch so fast, right? So they are not escaping. And they have nearly no teeth. But snakes that eat mice, they need venom to immobilize the prey, because if you bite a mice and it escapes, you will not find it again. Especially for snakes that eat birds, right? So if they bite the bird and the bird can escape, it will fly away. So it will not find its prey again. And that while these kind of snakes have a very prominent venom to immobilize it fast and quickly, and on the other side, they need strong teeth to keep it and not let it go. So by these structures, I can tell what happens. Okay. So this is a really ongoing scientific program that is huge, and a lot of scientists working on this field. So the major difference between vipers and elapids is that vipers have this maxilla, which is quite short, and then there's a venom teeth attached to it, and they have a completely mechanism in the end of the skull. So if I open the lower jaw, I activate 2 other bones that pressures the maxilla, and that makes the teeth coming up. So even in dead animals and dead specimens, if you open the mouth carefully, the whole mechanism would work and they will elongate the teeth. In elapid snakes, you not have this whole mechanism. So you have a long elongated maxilla, sometimes with small teeth at the end, and then you have a long elongated teeth at the front, which injects the venom. But elapid snakes, when they close their mouth, as they is having a standing teeth, it must be short. Otherwise, they will hit themselves when they close the mouth, right? That means that the length of the venom teeth has just a certain amount that they can do. Otherwise, they will not close their mouth. Vaporid snakes, because they have this mechanism, they can have really long teeth. Sometimes it's 30% of the whole skull, just one teeth. And when they open the mouth, it can flip off. And then you have this mechanism that give them the possibility to bite their prey. So these are really differences in this. But elapids and viperids are eating mostly the same kind of food, especially if you go to certain species. So all elapids eat mice, lizards, frog stuff, and there are species in the vipers that eat nearly the same. But these flipping Bauplan, this is the most difference. Yeah. So if you combine especially really closely related species towards each other, You can do it from the outside morphology, so you count the scales and you measure the specimen itself, how long is the tail, how long is the specimen, how big is the skull. But for the internal morphology, you sometimes can have more assumption on the lifestyle. So you, for example, can say by the vertebrae, if it is going to the ground, or if it is climbing a tree, or you have these famous flying snakes. So for the vertebrae itself, you can, for example, say if it is a ground living snake, or if it is climb up a tree, or you have these really famous flying snakes, which can make their vertebrae quite flat. And so you have a span of the skin that makes them flying or moving during the air. So for this, I can say, example, okay, this snake is doing this. This snake is living in this way. And this helps the taxonomics to distinguish between species, of course. Or I can say, in the skull, there are different kind of measurements or different kind of stuff that we find, especially if you have more than one specimen. So we had a famous project right now where we analyzed the lower jaw of closely related species. And we find differences to cell. Because you see this is this kind of species. Of course, it did not help someone in the field. But when you have skull remains, what you sometimes have from dead specimens, you can easily see which kind of species it is. And it gives hints towards where these specimens live, how to protect them, and which areas to protect. So doing my PhD now, I'm analyzing the venom glands, as I explained before. So we want to analyze what kind of mechanism is within the venom gland, and how did it inject the venom into its prey. So how many venom is produced before, how many venom is injected, and how it is working within the body. So it's really important to include also internal morphology into the scientific publication, because, you know, scientists are not gods. So sometimes you say, oh, I didn't see a different character, or I cannot analyze this, or in my opinion, they are all the same. And then there's another scientist coming maybe 5 years later, and he see the publication and the scans and say, yeah, but this character, this is completely different. So we have also the propose to public our data and to make it available for everyone else, that everyone else also can have a look at it. And there are good websites out there like Morphosource. Everyone can access it and can research for certain kind of characters, can research for certain kind of skulls, who is maybe interested in these kinds of. So we want to publish our data also that especially Nepalese scientists can access them for the species that are here occurring around. Yeah. This is a really broad question, so I can talk for hours. But I think to sum it up, once we have a CT scan, we can say, for example, this is the 3D model. And I have research questions also from artists. And they want to maybe have an exhibition about different kind of snake skulls, or they want to have an exhibition about bovid skulls itself. And when you have a CT scan of a really small specimen, let's say, so we have a good project with artists that's scanning amphibians. These are legless lizards living in the ground, and they have a skull from 5 millimeters in size, so really small. And when I do a good CT scan, I can zoom in like in a photograph. So we printed out these 5 millimeter skulls in 20 centimeter of size, just to give it to students, also to make education. Say, look, these are different kind of species, they live in different kind of grounds. This is how they look here, this is how they look in Turkey, this is how they look in Africa, so we make education with this, of course. On the other hand, you can say because the skulls are looking so different, you make like conservation areas, because you cannot take the guy from Turkey to Africa because he cannot live in the ground on this because of the skull shape there. So we can make also force the politics to say, see, they are looking so different, they are so different lifestyles. You have to protect certain kinds of areas for their living habitat. And then there's an artist coming say, hey, they are looking so cool and so nice. I want to print them out in glass and then illuminate it with some certain lights to make it like a fancy exhibition. We have also done this. And in the end, we also make like citizen science. So we say, look, when you go to your garden and you find a skeleton of something, make a picture from top, bottom, left and right side, and I can tell you by our CT scans what kind of specimens it is. So we have a combination of hardcore scientists, they want to describe new species, but also conservational political issues, coming up with artists and cool fancy stuff, back to education and citizen science. And this all with just one single CT scan. And in the moment, I have around 150,000 of them. So we can do broader ways. And last but not least, then the paleontologists come in place. So they say, I found a fossil somewhere looking exactly like the model that you created for this and this kind of species. So when I found a current living species right now, and I make a model of it, and I CT scan the fossil, then I can compare single bones of the fossil to the modern specimens. And for the most of the fossils, we can make analyses how old are these fossils. So if I do this, in the end I can say I have a species that is now coming from Kathmandu, for example, but the fossil is around 13 million years old. So I know that these species must living here for 30 million years. So I can also make a deep time analysis. How old are these lineages? How they are connected to each other? Sometimes, and mostly, they are longer on Earth than we are. So everything can be combined just with having these kind of analyzing structures. So there was a time that genetic people tell us that they know everything because they can do genetics. And then there was a time that morphologists say they can explain everything because they have the real specimen. In nowadays, we want to combine all of these fields, especially also with the internal morphology, as we realized that if you have a certain specimen that lives in a certain way, it adapted to its lifestyle. And then there's another kind of species that lives in the same way, so it adapted also to the same kind of way. Penguins are a really good example for this. So if you see a penguin wing, right, it looks like the fin of a fish because it goes into the water and dives through the water. But if you CT scan it, you will see that still the inner anatomy of this fin of this penguin looks like the wing of a bird. And that's how you know that penguins are birds, for example, is one character. So if a specimen adapted to a certain way of living, it adapted its whole anatomy and change also to this way of living. So if I want to go to the ground and dig into the ground, I have to make a really strong skull because I want to go into the ground. But the general Bauplan is not changing. It's just an adaptation. And this is what we see with CT scanning. And especially for species that are rare or highly protected, like the pangolin, for example, or like pandas. The red panda is a good example. You cannot go to the field and shoot a red panda and then rip off the skin and rip off the muscles to see the inner structures. So if you are lucky enough to have that specimen, you not want to destroy it. You want to make a CT scan to have the whole specimen preserved as it is, with fur and everything, and coloration. but you want to see the inner structure to realize if the red panda is really a panda bear or if it is something else, right? So this is how the CT technique gets more and more developed, and this is also how people are more interested in this, because we do not destroy any single specimen doing our analysis. Yeah, there are also a lot of interesting fields to tell. I think the one mostly is also the penguin that I already explained, where you analyze that it is a wing and not really a fin. In current statuses, you do mostly genetic analysis, and then you already have an idea where it comes in place. So the analysis of the inner structure and anatomy is not much used for taxonomic purposes itself. It's more to describe how specimens live, what they feed, so explaining more the ecology and the lifestyle of a specimen. So, of course, we use it also for certain characters to distinguish between the species, but the most impact we have is to explain how specimens live, what they're doing to protect them better. Right? If I know that a certain gecko lives on a certain way of walls, because the inner structure of the fingers and the scales that they have on the fingers made him glued to the wall itself, you know how to protect the environment, that you always have to protect these kind of walls to protect the species itself, and that it not can live on the ground or in a tree. And if you would destroy the habitat, you would destroy the species, because it cannot adapt that fast. Therefore, we need the CT technique to look inside, to define these characters, and to give the people an idea how to protect the environment and the lifestyle of the specimens. It's a hard question, as I'm the technical leader of the CT lab that we have. So I'm responsible for manipulating the machines to make the CT scans, but also to keep everything running, right? And to teach other students how to use this technique and machines. That means that the most of my working time is played with organizational stuff, technical stuff, education. The science that I'm doing in my free time. So on the weekends, I'm writing my papers. And when I come to holidays, I come here to Nepal to visit this beautiful place and find new species and understand also the living species. Because every specimen that we scan is dead. And I really want to understand how these specimens live in the field, right? To see the anatomy compared to the lifestyle itself. I know that in Nepal, the development of these kind of techniques is right in the beginning. So you not have the equipment laboratories so often here in this country. But what I would give an advice is to not give up. We also, 10 years ago, started with medical scanners. So try to find a medical institution that have good CT scanners and just test. Explain them why you want to do it, and you will realize that the most of the medical people, they are highly interested. So they really want to help. And dental CT scanners are not the best for the scientific purposes, but they also help to do. And once you have all the data sets, you need a good computer to analyze, of course. But you also can send messages to people that are already trained in this technique. So as I said, if you would send a message to our museum and say, hi, I want to analyze the common pit viper, do you have a CT scan of it? I also can use a pit viper that is in a collection somewhere else do a CT scan and send just the digital data. The only thing that the students really need is a high powerful computer to analyze this data. But we also do virtual trainings, so via Teams or Zoom, and then the people open the data on their own computer and say what I am supposed to do, and then I do this via a team chat. So a lot of my colleagues and I myself are really grateful to help. And once you have all this nice data and publication maybe, you can also force your institution to say, look, with the help of this and this persons, I did this in this publication, do you may think we can buy a CT machine by ourselves, because this is what could come up. I think that this development within Nepal and Kathmandu itself will be during the next years. So people have more understanding for the science itself. We met here in our holidays some amazing researchers that are interested in our fields. We teach them a lot, but we also learned a lot. And I think that this is a technique and a scientific research field that will develop within the next years. But you as Nepalese, you have to make the starting point, because as we Western people, we can tell you a lot, but you by yourself have to have started the interest in using these techniques. And I hope so that it will come up in the next years, because it's an amazing technique that helps you to analyze your data, to digitize your stuff, and to contribute in the bigger, wider sense of really good publicational stuff. For the next publications that we are doing, we include always genetics, morphology, and CT scanning. And what we get as a feedback is that this is something that also the scientific world really wants to see. And I think you should do this. But I really see the development, and I think that it will come up. In both, I have my highlights. So as I'm working in a CT lab, as I said, every specimen that I'm CT scanning is dead. So we are not working with living specimens. And here, the most interesting point that I have is to see the specimens alive, so I really enjoy the field work. Unfortunately, I not have so many holidays per year to come more often, but we spend more or less all of our holidays here to see the specimen alive, to find them, to understand the ecology. In the lab, I have really a bunch of different projects. So I'm scanning flowers in amber, 130 million years old. As I said, spiders, but also bovids. And all research projects that comes to my desk have their certain and really specific research interest. And sometimes we have seen new findings, and I always say we are the first persons, like in Star Trek, that see this character in 3D in a morphology. And sometimes you have this wow effect, because nobody had expected a certain character or a certain shape of something. And then we always sit in front of our computer and congratulate ourselves, because it's really the first time that someone sees a certain character. And this makes my life really amazing.
So, okay, at last but not least.
I think that the educational program that you have right now is really good here. And what I miss a bit, and what I think that has to be developed, is the cooperation with other parts. So I really see it often that people say, hey, I have this good and nice research ideas, but I do not know whom to ask how to develop a complete research project out of it. And I really would give the advice that these collaborations, these connections, that they just do it. Right? So you not have to be shy, you not have to be hidden behind others. You have an amazing species here in the country. You have a good research and educational plan. Just go out of yourself and send around messages and emails saying, hi, I have this and this idea, you have worked in this field, can you help me out with this project? And you will realize that a lot of other research scientists would like to help, because Nepal is a really nice country for doing herpetological research. But for us as a Western part scientists, it's sometimes also complicated to get the connection here. So we cannot just simply come and collect specimens and do our research. So also we need the connection. And what we have realized during our trips is that you want to work with us and we want to work with you, and nobody knows. So just come to us, just send a message saying, hi, this and this idea I have, what can we do? And then you will easily see that you get a network of people that really wants to do. And I think this could do the frustration rate quite low. And on the other hand, we could develop really amazingly projects. So always when we meet here other scientists, we also develop different kind of projects in our head. Now we go home and see how we realize it. But it always comes up, and that means for me that there are so many ideas out in the world. You just have to do it.
I got another one. Do you like to answer that?
Let's see.
It's a comparative or herpetofauna questions, Europe versus Nepal.
Okay.
Okay.
So in Europe, we have, especially for snakes, a really low diversity rate. So we have just, in Germany, let's say, we have just one really pit viper. And in whole Europe, it's really not much. And here in Nepal, as we visit different kind of stations during our trip, we realize that nearly every day you find a snake. Different kinds of course, and different species, but you have a so rich diversity that we miss in Europe, and it's poorly known on some places. So you have the locals that know their species and know their snakes and stuffs, but they do not talk to the scientists mostly, because they do not see why they should, because they know everything. But the scientists do not know. And in Europe, everything is more or less studied, so well known. But we have really poor diversity. And here you have it so enriched that I really hope for the future that there are more and more young scientists in Nepal comes up that study everything. And I give you a promise that if you do so, you will have known also your diversity much better. So it's really just a starting point, and I definitely think that within the next 10 years you will see how many species you have. It's incredible. So do the research here in the country.